Welcome to this chapter on uveitis and ocular inflammation. We'll begin by reviewing the basic concepts of immunology that are most relevant to inflammatory eye disease, with a particular focus on effector cells and the innate immune response. Understanding these early, nonspecific defense mechanisms provides the foundation for recognizing how ocular inflammation begins, how it is amplified, and how it can ultimately produce tissue damage within the eye. To begin, an immune response is a coordinated biological process designed to eliminate a harmful stimulus, whether that stimulus is infectious, foreign, or an altered component of the body itself. The clinical expression of this response is inflammation, which reflects activation at the vascular, cellular, and molecular levels. The immune system is therefore not a single organ, but an integrated network of tissues, cells, and soluble mediators. Its responses are broadly divided into innate and adaptive immunity, although these two systems function together rather than independently. Innate immunity provides immediate, nonspecific protection without requiring prior exposure. It serves as the first line of defense and helps shape the later adaptive response. Adaptive immunity develops more slowly, is highly antigen-specific, and generates immunologic memory, allowing a faster and stronger response when the same antigen is encountered again. Building on that overview, an immune response can be defined more precisely as a coordinated sequence of molecular and cellular events that protects the host from a wide range of threats. These threats include pathogenic organisms, toxic substances, cellular debris, and neoplastic cells. As we just introduced, immune responses are divided into innate and adaptive immunity. This division is useful for understanding their different roles, but it should not imply that they operate separately. In practice, the innate system detects danger and initiates the response, while also helping direct and shape the adaptive system. Together, these two arms create a coordinated defense that eliminates threats while attempting to preserve normal tissue. Now that we have defined the two arms of the immune response, we can compare them more directly. Innate immunity does not require prior exposure or immune education. It responds immediately and non-specifically, forming the first line of defense. As shown in the upper portion of the diagram, its major cellular components include neutrophils, basophils, eosinophils, mast cells, natural killer cells, macrophages, and dendritic cells. Adaptive immunity develops more slowly but is highly antigen-specific. It is driven primarily by lymphocytes, including B cells and T cells. B cells can differentiate into plasma cells that produce antibodies, while T cells develop into specialized populations, such as CD4 and CD8 T cells. An antigen is any substance capable of triggering an immune response, such as a toxin, foreign protein, or microorganism. The diagram also emphasizes that these systems communicate through cytokines and antigen presentation. Dysregulation of innate immunity tends to produce autoinflammatory disease, whereas inappropriate adaptive responses against self-antigens are more characteristic of autoimmunity. With that distinction in mind, we can now organize the major cellular components of the immune system. White blood cells, also called leukocytes, are nucleated cells responsible for immune defense. They are classified by features such as nuclear shape, the presence or absence of cytoplasmic granules, and their staining characteristics under the microscope. Leukocytes are broadly divided into myeloid and lymphoid lineages. The myeloid lineage includes neutrophils, eosinophils, basophils, mast cells, monocytes and macrophages, as well as dendritic cells, such as Langerhans cells. The lymphoid lineage includes T lymphocytes, B lymphocytes, and natural killer cells. This classification is important because each lineage contributes differently to innate and adaptive immunity, and each can play a distinct role in ocular inflammation. We'll begin with neutrophils, one of the major myeloid cells of the innate immune system. Neutrophils are polymorphonuclear leukocytes, or PMNs, named for their characteristically multi-lobed nuclei. As illustrated in the upper right, they also contain prominent cytoplasmic granules and lysosomes, which store the enzymes needed for rapid microbial killing. Neutrophils are the most abundant granulocytes in peripheral blood and are among the first cells recruited to sites of acute inflammation. Functionally, they are highly efficient phagocytes. They engulf pathogens and cellular debris, rapidly degrade that material, and amplify the inflammatory response by releasing granule enzymes and cytokines. This makes them powerful early effector cells, but their destructive contents can also injure surrounding ocular tissue when activation is excessive or poorly controlled. Neutrophils are especially important during the earliest phase of acute inflammation. They are rapidly recruited from the bloodstream into affected tissue through chemotaxis, meaning they migrate toward chemical signals released at the site of injury or infection. Because of this rapid response, neutrophils often dominate the inflammatory infiltrate in acute bacterial disease. In the eye, this includes bacterial conjunctivitis, keratitis, endophthalmitis, and some forms of scleritis. They may also be prominent in active viral infections, particularly herpes simplex keratitis and herpetic retinitis. Clinically, the presence of a neutrophil-predominant response should therefore raise concern for an active and potentially tissue-destructive infectious or acute inflammatory process. Moving next to eosinophils, these cells are identified by a bilobed nucleus and abundant cytoplasmic granules and lysosomes. Their granules contain basic or positively charged proteins that bind acidic dyes, such as eosin, which gives the cell its characteristic staining pattern. The key granule products include major basic protein and ribonucleases. These molecules are directly cytotoxic and are especially effective against parasites, which are often too large to be phagocytosed. Instead, eosinophils accumulate around the organism and release their toxic granule contents onto its surface. This mechanism is useful for host defense, but the same products can also damage surrounding tissue when eosinophilic inflammation is excessive. Functionally, eosinophils are most important in parasitic infections and allergic immune reactions. This is why peripheral eosinophilia can be seen in both settings. In ophthalmology, eosinophils are especially relevant at the ocular surface. They may be abundant within the conjunctiva and tears in allergic conjunctivitis, particularly in atopic and vernal conjunctivitis, where their toxic granule proteins contribute to epithelial injury and chronic inflammation. By contrast, eosinophils are not usually major effector cells in intraocular inflammation. The key exception is ocular infection with helminths, especially taxocariasis, where eosinophil-rich inflammation may develop in response to the parasite. We'll now turn to basophils and mast cells, which are closely related but occupy different compartments. Basophils circulate in the bloodstream, whereas mast cells are tissue-resident cells. Mast cells are broadly divided into connective tissue and mucosal subtypes, and their exact granule composition and function vary depending on the tissue in which they reside. Both cell types can respond rapidly by releasing pre-formed mediators stored within cytoplasmic granules. They can also synthesize additional inflammatory mediators de novo after activation. Their mediator profiles differ from those of neutrophils and eosinophils, which allows them to play distinct roles in vascular responses, allergy, and immune signaling. Although tissue location clearly influences mast cell behavior, the mechanisms regulating these differences are still not completely understood. Looking more closely at the two major mast cell subtypes, connective tissue mast cells contain abundant granules rich in histamine and heparin. When stimulated, they also synthesize prostaglandin D2, which contributes to vasodilation, vascular permeability, and recruitment of additional inflammatory cells. Mucosal mast cells behave somewhat differently. They contain less histamine at baseline and depend more heavily on cytokines derived from T cells for activation. Their major newly synthesized mediators are leukotrienes, particularly leukotriene C4. These differences illustrate how the local tissue environment shapes mast cell function and determines which inflammatory mediators are released. Mast cells are best known as key effector cells in type 1 hypersensitivity reactions mediated by IgE. Their surfaces express high-affinity Fc epsilon receptors that bind the constant, or Fc, region of IgE antibodies. When an allergen cross-links adjacent IgE molecules, the mast cell rapidly degranulates and releases its inflammatory mediators. Degranulation can also be triggered through other pathways, including complement activation and certain cytokines. This is important because mast cells are not limited to allergic disease. They also participate in cell-mediated immunity, wound healing, and non-IgE inflammatory responses. Therefore, mast cell activation may contribute to inflammation through both classic allergic and broader innate immune mechanisms. In the eye, mast cell distribution varies considerably by tissue. The normal conjunctiva contains numerous mast cells within the substantia propria, placing them close to blood vessels and environmental antigens at the ocular surface. In allergic and atopic disease, particularly vernal conjunctivitis, mast cell numbers increase not only within the substantia propria, but also within the conjunctival epithelium, which normally contains very few mast cells. This helps explain the prominent itching, redness, chemosis, and mucus production seen in allergic ocular disease. Connective tissue mast cells are also present throughout the uveal tract and may contribute to intraocular inflammatory signaling. In contrast, the normal cornea contains essentially no resident mast cells. Next are monocytes and macrophages, which represent different stages of the same cellular lineage. Monocytes circulate in the bloodstream as precursors. After entering tissue, they differentiate into macrophages, becoming larger and more functionally specialized. These cells contribute to both innate and adaptive immunity. They phagocytose pathogens and debris, release inflammatory mediators, and help activate lymphocytes through antigen presentation. In acute ocular infections, macrophages are often present even when neutrophils remain the dominant inflammatory cell. Monocytes are relatively large in the blood, measuring approximately 12 to 20 micrometers. After differentiating within tissue, macrophages may enlarge to as much as 40 micrometers, reflecting their increased capacity for phagocytosis and immune signaling. Macrophages within tissues come from two major populations. The first are tissue-resident macrophages, which are established during embryologic development and then acquire specialized functions based on the tissue they inhabit. Examples include Kupffer cells in the liver, alveolar macrophages in the lungs, and microglia in the brain and retina. The second population consists of blood-derived macrophages. These begin as circulating monocytes and migrate into mature tissues during inflammation, where they differentiate into macrophages. This distinction matters because resident macrophages help maintain local tissue homeostasis, while recruited macrophages are especially important in responding to injury and infection. Macrophages perform several complementary roles throughout the immune response. First, they act as sentinel cells, continuously surveying tissues for pathogens and signs of cellular damage. Once a threat is detected, they become effector cells by releasing inflammatory mediators, engulfing harmful material, and directly killing pathogens. Their function then shifts as the response evolves. Macrophages help regulate adaptive immunity, control the recruitment and movement of other immune cells, and promote tissue repair after the initial threat has been contained. This versatility allows the same cell population to initiate inflammation, sustain host defense, and later help resolve the response and restore tissue homeostasis. Macrophage activation occurs in stages. Resting monocytes can first be primed to function as antigen-presenting cells, allowing them to process foreign material and display antigen fragments to lymphocytes. Additional signals then drive full activation. Important activating stimuli include bacterial products such as lipopolysaccharide, the phagocytosis of pathogens coated with antibody or complement, and inflammatory cytokines such as interleukin-1 beta and interferon gamma. Once fully activated, macrophages produce maximal levels of inflammatory mediators and become much more efficient at killing and degrading the material they have engulfed. This is another key way macrophages connect the immediate innate response with the later adaptive immune response. With persistent stimulation, activated macrophages can undergo granulomatous transformation into epithelioid cells. These cells have large nuclei, abundant cytoplasm, indistinct borders, and a squamous-like appearance. Their presence is a hallmark of granulomatous inflammation. In uveitis, granulomatous inflammation may occur with infections such as tuberculosis, syphilis, herpes, fungal disease, or parasitic infection. It may also occur in non-infectious conditions, including sarcoidosis and granulomatosis with polyangiitis. Macrophages can additionally fuse to form multinucleated giant cells, which are seen in both granulomatous inflammation and foreign body reactions. Now let's talk about dendritic cells. These are bone marrow-derived professional antigen-presenting cells and serve as one of the most important links between innate and adaptive immunity. They recognize pathogens through pattern recognition receptors, including toll-like receptors. But unlike neutrophils and macrophages, their primary role is not direct pathogen killing. Once activated, dendritic cells increase the expression of co-stimulatory molecules, produce cytokines, and present antigen to T cells, directing T cell priming and differentiation. In the non-activated steady state, however, they can promote T cell unresponsiveness and help maintain immune tolerance. All dendritic cells express HLA class II molecules, which are essential for presenting antigen to CD4 T cells. Dendritic cells can be divided into several subtypes based largely on where they are found and how they enter tissues. Blood dendritic cells act mainly as circulating precursors for dendritic cells that will populate tissues or lymphoid organs. Migratory dendritic cells reside in peripheral epithelial tissues where they capture antigens. After activation, they travel through afferent lymphatic vessels to nearby lymph nodes. These are large cells with prominent cytoplasmic projections, or veils, that increase their surface area for antigen sampling and interaction with other immune cells. Resident dendritic cells, by contrast, arise within lymphoid organs directly from blood-borne precursors. Finally, inflammatory dendritic cells appear primarily during active inflammation and are often derived from recruited monocytes. Together, these populations allow antigen detection in tissues and efficient delivery of that information to the adaptive immune system. Langerhans cells are specialized myeloid dendritic cells that perform the same core antigen-presenting functions we just discussed. They are found within stratified epithelial tissues, including the epidermis, conjunctiva, and corneal epithelium, as well as other mucosal surfaces. They are recognized by their branching dendritic morphology and by characteristic intracellular structures called Burbeck granules. Unlike many recruited inflammatory dendritic cells, Langerhans cells originate during embryologic development from the yolk sac. Under resting conditions, they form a stable, self-renewing population that continuously surveys the ocular surface for foreign antigens and tissue injury. At rest, Langerhans cells function as relatively inactive antigen-presenting cells. Once activated by infection or tissue injury, they lose their characteristic Burbeck granules, migrate toward lymphoid tissue, and begin to resemble other activated dendritic cell populations. Their major functions include antigen presentation, promotion of delayed type hypersensitivity, and directing T cell differentiation. Within the conjunctiva and limbus, Langerhans cells are notable because they are the only cells that constitutively express HLA class II. In the normal cornea, they are concentrated primarily in the periphery. However, inflammation or stimulation of the central cornea can cause these cells to migrate centrally, increasing local antigen presentation and potentially amplifying the immune response. Having reviewed the major myeloid cells, we can now turn to lymphocytes, the principal cells of adaptive immunity. Lymphocytes are relatively small cells, typically measuring about 10 to 20 micrometers, with a large, round, densely staining nucleus and only a thin rim of cytoplasm. They arise from bone marrow stem cell precursors, but require further maturation within lymphoid organs before becoming fully functional. As shown in the diagram, lymphocytes differentiate into B cells and T cells. B cells can mature into antibody-producing plasma cells, while T cells develop into specialized populations, such as CD4 helper T cells and CD8 cytotoxic T cells. These subtypes are identified by their cell surface markers, which closely correlate with their immune function and molecular activity. Lymphocytes are broadly divided into three major categories: T lymphocytes, B lymphocytes, and non-T, non-B lymphocytes, which include cells such as natural killer cells. Each category has a distinct role within the immune response. As shown in the diagram, B cells can differentiate into plasma cells that produce antibodies. T cells can develop into specialized populations, including CD4 helper T cells and CD8 cytotoxic T cells. These broad groups can then be subdivided further according to the proteins expressed on their cell surfaces. Those markers are not merely labels. They reflect differences in how the cells recognize antigens, communicate with other immune cells, and carry out their effector functions. Although lymphocytes are generally associated with adaptive immunity, two lymphocyte populations also participate in rapid, innate-like responses. The first are gamma delta T cells, sometimes called sentinel T cells. These cells are commonly located within epithelial tissues, where they are also known as intraepithelial lymphocytes and can respond quickly to cellular stress or infection. The second group is natural killer, or NK cells, which belong to the non-T, non-B lymphocyte category. Natural killer cells can recognize and destroy infected or abnormal cells without requiring prior antigen-specific sensitization. Together, gamma delta T cells and natural killer cells blur the traditional boundary between innate and adaptive immunity, serving as an important bridge between the two systems.
Now that we have reviewed the major cellular players, we can summarize the defining features of the innate immune system. Innate immunity is broad-acting and rapid. It recognizes common non-self patterns, such as microbial proteins and lipopolysaccharides, without requiring prior exposure. This response is driven by germline-encoded receptors that detect features shared across many pathogens. Recognition triggers the production of biochemical mediators and cytokines, which recruit effector cells such as neutrophils and macrophages to the affected tissue. These cells then remove the offending stimulus through phagocytosis and enzymatic degradation. The key advantage of innate immunity is speed, although that rapid response is less antigen-specific than adaptive immunity.
The innate immune response does more than provide immediate defense. It also alerts and shapes the adaptive immune system through cytokine release, antigen presentation, and recruitment of additional immune cells. Those recruited cells then amplify and refine the response. Acute endophthalmitis provides a clinically important example. Bacterial toxins and damaged host cells trigger the rapid recruitment of neutrophils and monocytes into the eye. These cells release inflammatory mediators and phagocytose bacteria in an effort to control the infection. However, because the eye is a confined and delicate environment, this same protective response can produce substantial collateral tissue damage and rapid vision loss.
The innate immune system recognizes infection by detecting pathogen-associated molecular patterns, or PAMPs. These are conserved microbial features shared across broad groups of organisms, including components of bacteria and fungi, as well as characteristic viral nucleic acids. PAMPs are detected by pattern recognition receptors, or PRRs, located on the cell surface, within endosomes, or in the cytoplasm. Major receptor families include toll-like receptors, C-type lectin receptors, NOD-like receptors, and RIG-I-like receptors. Because these receptors occupy different cellular compartments and recognize different molecular patterns, their activation provides information about both the type of pathogen and whether it is located outside or inside the cell. Toll-like receptors are one of the major pattern recognition receptor families. Humans have 10 functional TLRs, and their location helps determine what type of microbial signal they detect. TLRs located on the plasma membrane primarily recognize components of extracellular microbes. These include TLR1 paired with 2, TLR2 paired with 6, as well as TLR4 and TLR5. In contrast, TLRs located within endosomes are positioned to detect microbial nucleic acids after pathogens are internalized. Important examples include TLR3, TLR7, and TLR9, which are particularly relevant to antiviral immune responses. The immune response is the biological process used to identify and remove an offending stimulus. When that response becomes clinically apparent, we describe it as inflammation. Inflammation produces 5 classic signs: pain, hyperemia, edema, heat, and loss of function. In the eye, these may appear as discomfort, redness, tissue swelling, and impaired vision or ocular function. Although these findings seem diverse, they arise primarily from 2 underlying physiologic changes: recruitment of inflammatory cells into the affected tissue and increased vascular permeability, which allows fluid and plasma proteins to leave the circulation. At the tissue level, inflammation is characterized by infiltration of effector cells and the release of multiple inflammatory mediators. These include cytokines, such as interleukins and chemokines, as well as lipid mediators, including prostaglandins, leukotrienes, and platelet activating factor. Activated inflammatory cells also generate reactive oxygen metabolites, including superoxide and nitrogen radicals, and release destructive granule enzymes such as proteases, collagenases, and elastases. In parallel, plasma-derived systems such as complement and fibrin become activated. Together, these mechanisms help eliminate the offending stimulus, but they can also produce substantial collateral tissue damage, particularly within delicate ocular structures. From a clinical perspective, immune activity is occurring continuously, usually at a level that produces no visible inflammation. The eye encounters routine insults every day, including allergens at the ocular surface and occasional microbial contamination, yet most are cleared without symptoms or tissue injury. Even during cataract surgery, small amounts of bacterial contamination may occur without progressing to infection because local defenses eliminate the organisms before the response escalates. The key point is that clinical inflammation does not simply indicate that the immune system is active. It reflects amplification of that response to the point that vascular changes, cellular recruitment, and tissue effects become clinically apparent. When immune regulation fails, the response can become clinically apparent as inflammatory disease. Autoimmune disease, specifically, results from a loss of adaptive immune tolerance to autoantigens, meaning normal components of the body are mistakenly recognized as targets. Autoreactive B cells may produce antibodies against self-antigens, while autoreactive T cells can directly injure tissue or amplify inflammation through cytokine signaling. The result is an immune attack against the body's own structures. A classic systemic example is systemic lupus erythematosus, in which widespread autoantibody formation and immune dysregulation can affect multiple organs, including the eye. In contrast to autoimmune disease, autoinflammatory disease is driven predominantly by dysregulation of the innate immune system. The inflammation develops without a primary pathologic antibody response or antigen-specific B or T cell attack against self-tissues. Instead, abnormal innate immune signaling leads to excessive cytokine production and recurrent or persistent inflammation. A clinically relevant example is systemic juvenile idiopathic arthritis, which has prominent autoinflammatory features and may be associated with ocular inflammation. The key distinction is that autoimmune disease is centered on maladaptive antigen-specific immunity, whereas autoinflammatory disease is centered on inappropriate activation of innate immune pathways. Rather than representing completely separate categories, most inflammatory diseases lie somewhere along a spectrum between autoinflammation and autoimmunity. Some conditions are driven predominantly by innate immune dysregulation, while others are more dependent on antigen-specific B and T cell responses. Many involve meaningful contributions from both. Ankylosing spondylitis is an example of a mixed spectrum disease, combining strong innate inflammatory pathways with adaptive immune features. More broadly, the cause of inflammatory disease is usually multifactorial. Genetic susceptibility interacts with environmental influences and, in some cases, infectious triggers. The resulting clinical phenotype depends on which immune pathways become dominant and which tissues are targeted. With the distinction between autoinflammatory and autoimmune disease established, we can now focus more specifically on innate immunity within the eye. A wide range of stimuli can activate this system, including infection, tissue injury, toxins, and cellular debris. Despite these different triggers, the response follows a common pattern. Innate immunity acts rapidly and nonspecifically as the first line of defense. These pathways ultimately converge on the recruitment and activation of innate effector cells, including neutrophils, macrophages, mast cells, and dendritic cells. The following slides will examine the mechanisms that initiate and coordinate this response. The major triggers of innate ocular immunity include both microbial signals and tissue injury. Bacteria-derived molecules can be recognized directly by pattern recognition receptors, while toxins or trauma can damage non-immune ocular parenchymal cells and release danger signals, even in the absence of infection. These signals activate the vascular endothelium, allowing circulating neutrophils to adhere, migrate into ocular tissue, and become activated. In parallel, innate signaling pathways recruit and activate macrophages. Together, these mechanisms convert local detection of infection or injury into a coordinated inflammatory response. These innate triggers ultimately produce three closely linked effects: recruitment of leukocytes, activation of those cells, and amplification of local inflammation. As summarized in the table, bacterial signals include lipopolysaccharide, other cell wall components, and secreted exotoxins. The response can also be modified by less specific soluble factors, including plasma-derived enzymes, acute phase reactants, and cytokines released by injured ocular parenchymal cells. Together, these signals transform a localized threat into a coordinated cellular response, while also determining how intense and potentially tissue-damaging that response becomes. One of the most important bacteria-derived triggers is lipopolysaccharide, or LPS, also known as endotoxin. LPS is an intrinsic component of the outer cell wall of gram-negative bacteria and is among the most potent activators of innate immunity. Structurally, it contains three major components: lipid A, the core oligosaccharide, and the O polysaccharide. Lipid A is the most biologically active portion and can activate immune effector cells at extremely low picogram per milliliter concentrations. Although the exact structure of LPS varies among bacterial species, its components are broadly recognized by the innate immune system, allowing rapid detection of gram-negative infection. LPS is recognized primarily through toll-like receptor 4, although toll-like receptor 2 may contribute in certain contexts. These receptors are expressed across multiple immune cell populations, including macrophages, neutrophils, dendritic cells, and lymphocytes. Once LPS signaling begins, it activates monocytes and neutrophils and increases production of inflammatory cytokines, such as interleukin-1, interleukin-6, and tumor necrosis factor. It can also promote degranulation, activate the alternative complement pathway, and stimulate vascular endothelial cells. Together, these effects rapidly recruit and activate inflammatory cells, but they can also intensify tissue injury if the response becomes excessive. Clinically, humans are intermittently exposed to low levels of gut-derived LPS, particularly during diarrheal illnesses or dysentery. This exposure has been implicated in dysentery-associated uveitis, arthritis, and reactive arthritis. At higher systemic levels, LPS is also a major driver of fever, septic shock, and widespread inflammatory responses. Experimentally, LPS is widely used to model innate ocular inflammation. In animals, injection of LPS into the foot pad can produce acute anterior uveitis, known as endotoxin-induced uveitis. This model is valuable because it isolates a predominantly innate inflammatory response and allows researchers to study the pathways that recruit leukocytes, disrupt the blood-ocular barrier, and produce anterior chamber inflammation.
Extending this concept to endophthalmitis, direct intraocular exposure to LPS produces a particularly intense inflammatory response. This helps explain why gram-negative intraocular infections are often especially aggressive. Bacterial replication is damaging, but the endotoxin itself also strongly activates innate immunity. Experimentally, intravitreal LPS causes a dose-dependent influx of neutrophils and monocytes. The inflammation is not confined to the vitreous, it also involves the uveal tract in retina. Therefore, much of the visual damage in gram-negative endophthalmitis reflects both direct bacterial injury and a powerful host inflammatory response affecting multiple ocular compartments.
LPS-driven inflammation begins when toll-like receptors on macrophages recognize the bacterial signal, leading to macrophage activation and release of pro-inflammatory cytokines such as interleukin-1, interleukin-6, and tumor necrosis factor alpha. One of the earliest histologic changes is platelet degranulation, which releases eicosanoids, platelet activating factor, and vasoactive amines. These mediators promote vascular permeability and leukocyte recruitment. As the response progresses, increasing intraocular levels of mediators such as leukotriene B4, thromboxane B2, prostaglandin E2, and interleukin-6 correlate with greater cellular infiltration and vascular leakage. The clinical implication is that the severity of endophthalmitis depends on more than simply whether viable bacteria remain in the eye. Studies comparing clinical isolates with genetically modified bacteria that produce fewer toxins show that toxin production strongly influences inflammatory cell recruitment and retinal cytotoxicity in both gram-positive and gram-negative infections. This means that antibiotic sterilization alone may not immediately stop tissue injury. Even after bacteria are killed, residual toxins can continue activating innate immune pathways, sustaining ocular inflammation, and contributing to vision loss. Therefore, the clinical course reflects three interacting factors: bacterial viability, the remaining toxin burden, and the intensity of the host inflammatory response. Beyond LPS, many other bacterial cell wall and membrane components can also activate innate immunity. These include polysaccharides, lipids, and proteins that are recognized even without a pre-existing adaptive immune response. Importantly, viable bacteria are not always required. Killed lysates from gram-positive bacteria and mycobacteria can directly activate macrophages, which is why some of these materials are used as immune adjuvants. Similar bacterial components have also been used to induce experimental arthritis and uveitis. Although the specific molecules vary, many of the downstream pathways resemble LPS signaling, ultimately producing cytokine release, leukocyte recruitment, and local inflammation. Exotoxins differ from structural bacterial components because they are actively secreted into the surrounding environment. Many function as enzymes that directly disrupt cellular processes. These toxins are often not inflammatory by themselves. Instead, they injure tissue, cause cell death, and release danger signals from damaged host cells. The immune system then responds to that destruction, producing secondary inflammation. The key concept is that exotoxin-associated inflammation may be driven less by direct immune recognition of the toxin and more by the tissue damage the toxin creates. Specific exotoxins illustrate how bacterial products can produce both direct tissue destruction and secondary inflammation. Collagenases degrade the extracellular matrix, leading to structural breakdown. Hemolysins, such as streptolysin O, can kill neutrophils and release their destructive granule contents into surrounding tissue. Phospholipases disrupt cell membranes, producing rapid cell death and necrosis. This is especially important in Bacillus cereus endophthalmitis. Its hemolysin BL toxin can cause direct retinal necrosis and retinal detachment, and even a very small bacterial inoculum may produce complete retinal functional loss within hours. Therefore, the severity of these infections reflects not only bacterial growth, but also rapid toxin-mediated retinal injury and powerful activation of the innate inflammatory response. Beyond microbial products, tissue damage itself can directly activate innate immunity. Non-immune ocular cells are not passive bystanders. Cells of the iris and ciliary body epithelium, retinal pigment epithelium, Mueller cells, and the corneal and conjunctival epithelium can all detect injury. Triggers include toxins, mechanical trauma, and surgical or laser injury. In response, these cells produce cytokines, eicosinoids, and other inflammatory mediators that recruit innate effector cells, particularly neutrophils and macrophages. This explains why significant ocular inflammation can occur even without infection. Damaged tissue can initiate the same core inflammatory pathways. These examples show how non-immune ocular cells can serve as active sentinel responders. Corneal epithelial cells can phagocytose staphylococci, while contact lens-related microtrauma can stimulate the ocular surface epithelium even before overt infection develops. Similarly, mechanical chafing from an intraocular lens can activate the iris or ciliary epithelium, and retinal laser treatment can trigger inflammatory signaling through controlled tissue injury. The common principle is that damaged or stressed ocular cells do not simply wait for leukocytes to arrive. They initiate the response by releasing mediators that recruit and activate innate immune cells, thereby amplifying local inflammation. A clear clinical example of damage-driven innate inflammation is uveitis, glaucoma, hyphema, or UGH syndrome. This occurs after cataract surgery when an intraocular lens or its haptics repeatedly rub against the iris, ciliary body, or angle structures. It was classically associated with rigid anterior chamber lenses, but it can also occur with posterior chamber lenses, particularly when a one-piece lens is placed in the sulcus or the haptics are malpositioned. The repeated mechanical trauma stimulates local cytokine and eicosinoid production, increases vascular permeability, and permits plasma-derived factors such as complement and fibrin to enter the eye. Contact with the lens surface may further promote immune activation, including low-grade foreign body responses or adherence of inflammatory cells. Clinically, this produces the characteristic combination of recurrent uveitis, elevated intraocular pressure, and hyphema. Now that we have seen how tissue injury activates innate immunity, we can examine how neutrophils are recruited from the circulation. Neutrophils may exist in resting or activated states, and their movement into inflamed tissue is rapid but tightly regulated. Recruitment involves two major events. First, neutrophils adhere to activated vascular endothelium through cell adhesion molecules, particularly within postcapillary venules. They then transmigrate across the endothelial barrier and extracellular matrix, following chemotactic signals toward the site of injury or infection. Endothelial activation can be triggered by LPS, physical injury, thrombin, or histamine, preparing the vessel wall to capture circulating neutrophils and direct them into the affected ocular tissue. Neutrophil recruitment proceeds through a coordinated sequence of rolling, firm adhesion, and transmigration. Initially, selectin-mediated interactions create weak, reversible attachments that slow the neutrophil and allow it to roll along activated endothelium. L-selectin is expressed on the neutrophil, while E-selectin and P-selectin are induced on endothelial cells. Local cytokines, platelet activating factor, and bacterial products then activate neutrophil beta integrants, including LFA1 and MAC1. These integrants bind endothelial ICAM molecules, converting rolling into firm adhesion. The neutrophil can then cross the endothelial barrier and migrate toward the inflammatory stimulus. Importantly, these early recruitment events can begin within minutes of tissue injury or infection. After firm adhesion and transmigration, neutrophils move through tissue by chemotaxis. Short-range signaling molecules form concentration gradients, and the neutrophil migrates toward the highest concentration. Important chemotactic signals include complement fragment C5a, fibrin breakdown products, substance P, bacterial formal peptides, leukotrienes, and interleukin-8. The diagram summarizes the full sequence from rolling to integrin-mediated adhesion to transmigration and finally activation near the pathogen. Activation begins during adhesion, but becomes strongest at the site of injury or infection, where bacteria, LPS, complement, leukotrienes, and platelet activating factors stimulate the cell. Neutrophils then remain locally, perform their antimicrobial effector functions, and ultimately die rather than returning to the circulation.
Once neutrophils reach the site of infection, they eliminate pathogens through phagocytosis. This is a receptor-mediated process, with antibody Fc receptors and complement receptors serving as the two major recognition systems. Phagocytosis becomes much more efficient when the pathogen has been opsonized, meaning it is coated with antibody or activated complement fragments. These coatings bind to their corresponding receptors on the neutrophil surface and trigger engulfment. The cell membrane then extends around the pathogen and encloses it within an intracellular compartment called a phagosome, where the next stages of microbial killing and degradation can occur.
After the pathogen has been enclosed within a phagosome, cytoplasmic granules and lysosomes fuse with it to form a phagolysosome. This compartment concentrates antimicrobial polypeptides, reactive oxygen species, and reactive nitrogen species, which work together to kill and degrade the organism. These mechanisms are highly effective, but they are not completely contained. Lysosomal enzymes and reactive radicals may escape into surrounding tissue, where they amplify inflammation and cause collateral injury. In the eye, this trade-off is clinically important. The same neutrophil response that controls infection can also damage delicate structures such as the cornea, retina, and uveal tract. Neutrophils carry several antimicrobial products within their cytoplasmic granules, which they release into the phagolysosome or surrounding tissue after activation. Major components include bactericidal permeability increasing protein, or BPI, defensins, lysosome, lactoferrin, and serine proteases. These molecules attack microbes through complementary mechanisms. Some disrupt bacterial membranes, others degrade structural components, and lactoferrin restricts growth by binding iron. Together, they make neutrophils highly effective antimicrobial effector cells. However, as we discussed on the previous slide, release of these products outside the phagolysosome can also injure nearby ocular tissue. Neutrophil granules also contain potent inflammatory enzymes, including elastase, myeloperoxidase, and matrix metalloproteinases, such as gelatinase and collagenase. While these products remain membrane-bound within the granule, they are relatively inactive. They become active and soluble when the granule fuses with a phagosome or with the neutrophil's plasma membrane. This activation is essential for microbial killing, but extracellular release can rapidly damage surrounding tissue. In the cornea, collagenases and matrix metalloproteinases can degrade stromal collagen and produce liquefaction or corneal melting. This contributes to tissue destruction in bacterial keratitis and scleritis, is especially severe in Pseudomonas infection, and also plays a major role in rheumatoid arthritis-associated peripheral ulcerative keratitis. This concludes our overview of the cellular components and innate immune mechanisms involved in uveitis and ocular inflammation. The central theme is that inflammation is protective when appropriately regulated, but the same cells and mediators that eliminate infection can also cause significant collateral damage within delicate ocular tissues. Understanding that balance provides the foundation for interpreting inflammatory eye disease and for targeting treatment appropriately. Thank you for watching.